Hi everyone, Dr. Rashid here. In this video, I will be explaining you the structure of DNA molecule. And all of you know regarding DNA. DNA is the storehouse of genetic information in all organism. Coming to the structure of DNA, DNA is made of deoxyribonucleotide. DNA is a polymer of deoxyribonucleotide, and this DNA structure is made of two such deoxyribonucleotide strands. So DNA is a double-stranded molecule, and this double-stranded molecule twisted into a helix. So DNA is a double-stranded helix, and we can compare the structure of DNA with a spiral staircase. And this structure was first described by James Watson and Francis Crick in the year 1953. So DNA is a double-stranded helix, and the structure is almost similar to a spiral staircase. So we can compare the structure of DNA with a spiral staircase. So these handrails and these steps. So in case of DNA, these are the handrails which are made of phosphate group and the deoxyribose sugar. And the steps of this staircase, it is made of nitrogenous base, either purine or a pyrimidine base. So the backbone or the handrails, it is made of phosphate and sugar molecule, and the steps are made of the nitrogenous base. So coming to the features, salient features of DNA structure, the first point is it is a right-handed double helix. That is the direction of this double helix. It is a right-handed double helix, and these two polydeoxyribonucleotides twisted to form a helix. And this spiral strand is composed of sugar phosphate backbone. So this backbone is made of sugar molecule with phosphate group, and the central part, that is the steps of this staircase, is made of this nitrogenous base. Coming to the second point, that is the base pairing rule. So the two strands are complementary to each other. So adenine, which is present on one strand, form hydrogen bond with thymine, which is present on the other strand. Similarly, guanine present in one strand form hydrogen bond with cytosine on the other strand. That is, they follow base pairing rule. So in one strand there is adenine, and this adenine form hydrogen bond with thymine, and guanine, which is present on one strand, they are paired with cytosine on the other strand. So one side it is purine. This is purine bases, and purine bases form hydrogen bond with pyrimidine bases on the other strand. So number of purine is always equal to number of pyrimidine, or we can say adenine plus guanine is equal to thymine plus cytosine. That is known as Chargaff's rules. So Chargaff's rules means the number of purine which is always equal to the number of pyrimidine. It is applicable in case of a double-stranded DNA molecule. Coming to the third point, that is hydrogen bond. That is the interaction between these nitrogenous bases. So purine form hydrogen bond with pyrimidine, corresponding pyrimidine, and adenine form 2 hydrogen bond with thymine, whereas guanine form 3 hydrogen bond with cytosine. So guanine cytosine bond is stronger than adenine thymine bond. So adenine form 2 hydrogen bond with thymine, and guanine form 3 hydrogen bond with cytosine. The fourth point, that is the direction of the each strand. So in one strand it is directed in 5' to 3' direction, whereas in other strand it is directed in 3' to 5' direction. So we can say the direction is anti-parallel. The two strands run in anti-parallel direction, and the spatial relationship between the two strands create some depressed region within the DNA molecule, and these depressed regions are known as the grooves. So there are two types of grooves, that is major grooves and minor grooves. So this one is the major grooves, and this one is the minor grooves. So what is the importance of these grooves? And the functions of DNAs are regulated with the help of some regulatory protein. And this regulatory protein interact with this DNA molecule through this major and minor grooves. So grooves are the site of interaction of DNA with the regulatory protein. So DNA is having helix configuration, and this represents one turn of the helix. One turn of the helix, and this distance is known as the height of the turn or pitch of the helix, and the height of the turn or the pitch of the helix is around 3.4 nanometer, and this turn contain base pair, and number of base pair present in each turn is around 10, and this distance between this base pair is 3.4 divided by 10 nanometer, that is 0.34 nanometer. So the height of the turn is around 3.4 nanometer, and one turn contain 10 base pair, and the distance between the base pair is around 3.4 nanometer, and this distance, that is known as the diameter of the helix, and the diameter of the helix is around 2 nanometer. So I repeat, the diameter of the DNA double helix is around 2 nanometer, and pitch of the helix or distance of one turn is around 3.4 nanometer, and one turn of the DNA double helix contain 10 base pairs, and distance between two adjacent base pair, it is around 0.34 nanometer. So each helical turn contain about 10 base pairs, and pitch of the helix or the height of the turn, that is around 3.4 nanometer, and distance between two adjacent base pairs, it is around 0.34 nanometer, and the diameter of the DNA double helix, it is around 2 nanometers. So this is the structure of DNA, that is one strand. So DNA structure may contain two deoxyribonucleotide strand. That central part is contributed by the bases, that is non-polar bases represents the central core of the DNA molecule, and the peripheral part of the DNA molecule represents the polar or hydrophilic molecule, that is the phosphate and the deoxyribose sugar. And adenine always paired with thymine through 2 hydrogen bond, and guanine always paired with cytosine with 3 hydrogen bond. And coming to the different types of DNA, there are 6 types of DNA, that is A, B, C, D, E and Z. So there are 6 types of DNA, and A, B, C, D, E, all these are right-handed helix, and Z form, Z DNA, that is only left-handed DNA. Among these 6 types of DNA, the most common form is B form. So B form of DNA is the most common form of DNA. That is the type of DNA which is seen in normal physiological condition. And in B form, one turn contain 10 base pairs. And in case of A form of DNA, one turn contain 11 base pair. And in Z form of DNA, one turn contain 12 base pairs. So that's all regarding the structure of DNA. Thank you.